Ko tēnei Vivian Hunt, kia koutou katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, I would like to welcome you to the first of our monthly food science seminars spotlight, when we will bring up the latest topic on food safety science. Today, I'm delighted to have introduced Dr. Kath McLeod, who's the Director of the New Zealand Food Safety Science and Research Centre. Some of you will know Kath from her previous days of working at MPI, and she was called Kath Seema. Kath, we've been very lucky to bring Kath back to New Zealand recent times, and she joined the centre as the director of the centre in December last year. Kath has had an extensive experience in food safety science. She led interdisciplinary food safety research programs that have combined molecular biology, microbiology, chemistry and stats, and risk assessment both in New Zealand, Australia, France and the UK. She's held diverse roles with regulatory, scientific and political sectors, which has given her a lot of insights into key drivers and challenges faced by each of the groups in the food safety industry or food industry. Kath is particularly skilled at working with industry and regulatory groups to identify food safety and market access issues. She has a PhD on foodborne viruses from the Victoria University of Wellington, which was funded by ESR, postgraduate scholarship, and she's led postdoctoral research, both focused on norovirus and hepatitis A in both produce and seafood. Now, before I do hand over to Kath, I just wanted to take you through some of the webinar housekeeping. This is new to us, and we have around 50 people joining us today. And so to make sure we don't overload Zoom, we want to just have everyone dial in on audio. And we ask you to put yourself on mute so there's no background noise. And the best view, Kath, as she talks, if you present, here's what you should be looking at. Go to speaker view and set that to speaker view, not gallery view, so you can see Kath speaking and sharing her slides. Then if you have questions, if you can send them to me, just select my name on the chat and send me questions during the presentation, and I'll moderate some of those talk. So the next seminar is going to be held on it's on the last Friday of every month at 10 AM, and the next one will be 7th of March. Topic to be finalised, but we'll advertise that in the next week or so. So thanks, Kath, for doing this first talk, and I'll hand over to you now. And please remind everyone to go mute.
Thanks, Vivian. No mai, haere mai, ngā mihi mahana, kia koutou. A warm welcome to you all, and thanks for dialing in today. As Vivian said, I'm currently the Director of the New Zealand Food Safety Science and Research Centre. The centre is a unique partnership of industry. So we have the poultry industry, horticulture, seafood, meat, dairy, and government, including New Zealand Food Safety and MPI, and research experts in the form of 7 research organisations. It's a special partnership, and it has the explicit goal of trying to reduce risk related to food safety issues. So today I've got the honour of talking to you about a really important food safety issue, and that is foodborne viruses. And I quite like this quote from Bill Marler: talking about food safety does not make your food less safe, it makes it safer. So I hope that our chat today about foodborne viruses promotes risk reduction in that respect. So a quick overview of today's discussion. What is the impact of foodborne viruses in New Zealand? What are the main viruses of concern? How do foods become contaminated? What test methods are used? And how can we reduce risk? So firstly, to focus on foodborne viral illness and the impact of that. What is the virus contribution to foodborne illness in New Zealand? Norovirus is the most common pathogen implicated in outbreaks. So in over 40% of reported outbreaks in 2007, norovirus was the confirmed cause. Most outbreaks, however, are transmitted person to person, and expert elicitation in New Zealand has suggested that about 2.7% of those are foodborne. So it's a pretty significant pathogen that we should be thinking about. So just looking at this in a different way, this is a graph taken from the ESR annual summary of outbreaks, and it shows the pathogens implicated in disease outbreaks from 2008 to 2007, and the dark blue bars down the bottom show the proportion of outbreaks attributed to norovirus. The balance of those bars are made up of other pathogens and predominantly bacterial in nature. So how many people actually get sick from foodborne viruses in New Zealand? And that's a really difficult question to determine. There is significant underreporting, and that relates to the fact that a lot of us don't report to a doctor if we get a short-lived case of gastroenteritis. There has been some work done in the UK on this, and that work suggests that for every case reported to surveillance system, there are 288 additional cases. So that gives you some idea about the scale of underreporting that's going on. And then if we look at a different part of the world, the European Union, again, viruses are the leading cause of foodborne disease in Europe and have been since 2014. In 2014, they were responsible for about 20% of the outbreaks, with over 1,000 being caused by foodborne viruses. And the major viruses implicated are norovirus, which was responsible for 98%, and hepatitis A at just under 2%. Looking at some specific outbreaks, this is an outbreak that occurred in New Zealand in 2006. It evolved around 115 cases of norovirus, and it was related to the consumption of oysters in the corporate boxes at a rugby match of New Zealand versus Ireland. I think the caterer ran out of New Zealand grown oysters and substituted them with raw imported Korean oysters. The Korean oysters did say to cook them prior to consumption, however the caterer didn't do that, and illnesses ensued. I think the important thing to note is that the All Blacks won on the day. So another outbreak in 2002 occurred in New Zealand. This outbreak was a hepatitis outbreak related to raw blueberry consumption. There were 81 cases of hepatitis lab confirmed in this outbreak, and it was traced back to a single orchard in the North Island of New Zealand. The investigation on farm found hygiene failures. There were pit latrines at about 4 feet below the surface of the ground, and there had been significant rainfall in the preceding weeks. And so the implication was that sewage from those pit latrines may have contaminated the area. There was also no running water, soap or towels. And in more recent times, some of you may be more familiar with this outbreak, which occurred in 2015 and involved 7 cases of hepatitis in New Zealand, and it stemmed from frozen imported berries, and China was implicated. New Zealand Food Safety, on the back of this, put out advice about boiling berries for a short period of time to ensure their safety. And indeed, New Zealand Food Safety aren't alone in that respect. Similar advice exists in other parts of the world. And this is advice that the Food Safety Authority of Ireland have on their website about boiling imported frozen berries. And I should say I had a little bit to do with Ireland when I was over in the UK, and did some work with Safe Food, which is an Irish food safety organisation. And this advice really stemmed out of a problem that occurred in 2003, 2014, and Ireland had 21 cases of hepatitis A. As it turned out, the 21 cases in Ireland were really the tip of the iceberg, and it was an EU multi-state outbreak. This graph shows the course of the outbreak over time in 2013 and 14, and the number of cases in each country, and you can see that Italy really was the epicentre. In total, there were around 1,500 cases confirmed, with 2 deaths. And I think, just a really pertinent point here was that the traceback was extremely complicated, and this is due to the berries, the fact that berries are used as ingredients in a large number of products. So the traceback involved 2,000 companies approximately in the production, processing and retailing aspects, and it wasn't possible to boil it down to a definitive source, although there was suggestions that the virus was introduced in the freezing process in Bulgaria for blackberries and through Polish red currants. So it's an interesting outbreak. The cost of these outbreaks is high. So this is an example from Wallace Lake in Australia in 1997. Wallace Lake is a very productive oyster growing area. And in 1997, it was found that the oysters in this area were contaminated with effluent from a myriad of sources. There were 500 hepatitis A cases, 70 were hospitalised, and one person unfortunately died. And the cost of this outbreak was estimated by an economist in subsequent years. The national health cost was estimated to be 12.1 million. Oyster sales fell over a number of years by 20%, and this resulted in 60 less workers employed in the oyster industry in the area. And the fishing industry was also impacted over a number of years. So this is the wider fishing industry around Wallace Lake, where they lost up to 30% market value because people simply stopped eating seafood in general from this area. And there was a tourism impact that was estimated to cost around 1.1 million. So the costs are high, and it's always better to prevent these things. So what are the main viruses of concern? So this again is a table from the ESR annual summary of outbreaks in 2007, and it shows hospitalised outbreak cases. So we can see here that norovirus is the head of this table, so it really is the virus that we're most concerned with, followed by hepatitis A with much lower impact. There's also some other viruses that can be involved from time to time, and here you see rotavirus, adenovirus featuring. Looking at this a different way, this is a review undertaken by Bello et al. In 2013, and this was a systematic literature review of global shellfish-borne virus outbreaks. And we can see here that the vast majority, over 97%, are caused by norovirus and hepatitis A, and that to a much lesser extent other viruses feature. It is acknowledged that this review is probably biased because because not all countries publish this type of data. However, it does give a bit of a picture on what's going on. So before I get into the detail on norovirus and hepatitis, I just wanted to touch on a couple of other viruses of interest. And the first one is hepatitis E virus. Hepatitis E is a zoonotic virus, and it usually is a mild disease, but it can be life-threatening for at-risk populations, including those that might have solid organ donations. So the interesting thing is that in Europe, it's been observed that hepatitis E infections have increased over the last decade. So you can see here there's been 21,000 cases of hepatitis E reported between 2005 and 2015. This has prompted a lot of attention, and so in 2018 the Food Standards Agency convened a workshop on this matter. And it's fair to say that for at least genotype 3, hepatitis E genotype 3, that the main transmission route is considered to be foodborne, primarily through pigs, but deer are also implicated as well. There is an implication that other foods may play a role in transmission, but the extent to which they do that remains very uncertain. Hepatitis E has also been looked at in New Zealand by New Zealand Food Safety and ESR, and this is a graph from an article published by Nicola King from ESR, and it shows the transmission routes for hepatitis E. The solid lines are known transmission, the dotted lines are where we're less certain. So you can see we're quite certain about pigs and deer and less certain about shellfish and food produce and their role in transmission. In New Zealand, in terms of hepatitis E, there have been 25 cases reported to the public surveillance system between 2008 and 2016. However, it's likely to be underreported because mostly it's a mild illness. And there was a 2014-15 survey of over 1,000 blood donations that found 10% seropositivity. So there's a suggestion there that its impact may be much wider than what we really understand. And I think it would be remiss today, given the coronavirus situation, not to touch on this, albeit very briefly, given the severity in China and other countries around the world. The coronavirus that's circulating at the moment ranges clinically from mild respiratory syndrome to life-threatening pneumonia. And the case fatality rate at the moment is around 2%. That's more than influenza H1N1 in the 2009 pandemic, which was less than 1%, but less than the SARS epidemic in 2002-03, which was around 10%. The epidemiological evidence to date suggests zoonotic transmission from Wuhan's seafood market, where wild animals are traded. But I have to say this is somewhat speculative at the moment. Molecular evidence suggests the virus is most closely related to a coronavirus from a horseshoe bat from the Yunnan province. These bats are understood to be hosts for SARS-related coronavirus, and COVID-19 may have emerged in a similar way to SARS. However, foodborne transmission is pretty unlikely. It's a respiratory virus that's transmitted from person to person, and it's spread when an infected person talks, coughs, or sneezes small droplets into the air. There is no evidence to date that foodborne transmission is occurring. And if people are interested in that aspect, the FSAI have on their website some reasonable guidance for food producers around this matter. So moving on to the main viruses of concern here. What is norovirus? Well, it's an RNA virus with a protein coat. It replicates in human intestine cells to produce gastroenteritis. It's very hardy. It survives for at least 1 week on stainless steel and other surfaces. And it has a very low infectious dose. There has been one estimate, one paper published that suggests that as little as 1 viral particle has a probability of illness of 50%. So it's very, very infectious. However, there's some international speculation on whether this really overestimates the infectivity of norovirus. This graph shows some data from Lee's 2005, and each individual bar on the graph represents an oyster sample and shows the number of viral genome copies in that sample for norovirus. The black bars show samples that were associated with outbreaks. So what you can see here is that the higher the viral numbers in these shellfish samples, the more likely it is to be linked to an illness outbreak. So I think there's more work that's needed in this space for sure around dose response. In terms of clinical symptoms, the virus is characteristic of a very violent gastrointestinal illness with diarrhea and vomiting and generally projectile vomiting. So the incubation period is quite short at 1 day, and the duration is generally 1 to 2 days. The little graph on here shows the viral concentration of G1 and G2 on the left and right respectively in fecal samples, and you can see that concentrations range from 10 to the 4 to 10 to the 11 per gram. And so when you overlay that with the infectious dose information, you can see why this virus is so readily transmitted in the community. Norovirus has high genetic diversity. G2 strains predominate. So those are the strains on the left. And we now understand a little bit about how the virus infects humans, and histoblood group antigens play a role here. So norovirus specifically recognise these antigens, and you guys probably remember them as ABO, the blood type antigens, but there are also others. And different strains of norovirus bind to different antigens. So a single strain can't infect everybody, but there is a strain for everyone. So moving on to hepatitis A virus, like norovirus, it's transmitted via the fecal-oral route. It has a longer incubation period though, so from 15 to 50 days, and this makes it very difficult to pinpoint causative foods, as people have generally forgotten what they've consumed in that far behind in history. Illness usually lasts for a couple of months. The symptoms are also gastrointestinal, but can also become more severe than that. It is vaccine preventable, and I think a really interesting aspect of hepatitis A is that disease is more severe in adults than children. So it's problematic in developed countries like New Zealand where immunity is low. So in New Zealand we have a low prevalence of HIV, around 5 outbreaks identified in 2017 with 20 cases. This is a little map put together by the CDC in the US, and it shows anti-HIV prevalence. So you can see New Zealand and Australia and much of Northern America and Europe have a very low prevalence. And this puts us somewhat at risk of consuming foods from highly prevalent areas, contaminated foods from highly prevalent areas. So how do foods become contaminated? Handling and water are the key transmission pathways. So failure in hygiene or contact of food with human feces or vomit that occurs through either poor handling or contaminated water, and it can occur through the supply chain at production, processing, and at the food preparation step. And foods consumed raw are more at risk of viral contamination because there's no kill step. So these are bivalve shellfish, berries, leafy greens, and bakery products. This graph is a graph from an EU report on zoonoses and foodborne outbreaks in 2014. And you can see here that the proportion of foodborne viral outbreaks related to shellfish constitutes the largest number of outbreaks at around 36.8%. The red and the blue segments here represent fruit and vegetables, so they comprise around 20% of outbreaks, so they're quite significant too. But things like bakery products also feature. So what's the major shellfish contamination pathway? Well, it's fairly simple. It's sewage into seawater in which shellfish grow. So that can happen via a number of mechanisms through boats in which onboard treatment systems may or may not be used. And if they do have treatment systems, they may or may not work 100%. There have been cases in the past overseas of discharge from shellfish harvesting vessels, which has caused contamination of production areas. Urbanisation is also an issue for shellfish with the drift of populations to coastal areas. Everyone wants to live by the sea. And sometimes the effluent systems in those areas are not suitable to support such large increases in the population. So on-site systems and communal systems need to be adequate for the size of the populations. Failure of those systems obviously increases risks to the public health of the community. How long do the viruses remain infectious for? So once the shellfish are contaminated, what sort of period of time are they an issue? Is there a potential public health issue? So studies on HIV suggest that while the genome can be detected for 6 weeks, the virus is infectious for around 3 weeks. In the US, 21-day closures are implemented for sewage spills, and it seems to be protective of public health. Looking at norovirus, this is some results from a paper published by Brake et al. In 2017. There was a study undertaken in the Brunswick River in northern New South Wales, and Felicity studied how long norovirus persists after a sewage spill and how far it travels. And she found that she could detect the virus by PCR for up to 41 days and that it travelled over 5 kilometres. So why does it persist for so long? Well, we now know that these histo blood group antigens are very evolutionarily conserved, and they're actually present in oysters and other food types as well. So we know that norovirus binds specifically to histo blood group-like antigens in the Pacific oyster stomach, particularly to A antigen. Moving to produce. How do produce become contaminated? Well, they can become contaminated at multiple steps in the supply chain. At production, through contaminated irrigation water, or through harvesters' hands. At processing, through rinsing water or food handlers' hands, and they can become contaminated at the point of sale through consumers themselves. And similar to oysters, we now know that norovirus binds to histo blood group-like antigens in lettuce and other fruit and vegetables. So if, for example, irrigation water is contaminated with viruses, there is potential that these viruses can get taken up through the vascular system of the plants and specifically bind to these antigens. And this means that washing with water may not always be effective. What test methods are used for norovirus and HIV in shellfish and produce? So since 2012, there has been an ISO standard method for norovirus and hepatitis A. And this is what's called a horizontal method, so it includes a number of matrices which include shellfish, salad vegetables, and soft fruits. So the basic components of the method are sample collection, viral elution, viral concentration, extraction of the nucleic acid, and then amplification by polymerase chain reaction and qPCR detection. There are a number of controls in the method, positive and negative controls, and controls that enable quantification of the amount of virus. So the pros and cons of the method are, there are detailed protocols available, and this makes it quite easy for labs to adopt the method. And it's possible to compare and evaluate labs from different results from different labs, and there are interlab studies that are run for this method now. There are also commercial kits based on this method available, making it quite easy to adopt the method. And I think an important point here is that commercial labs overseas, particularly in Europe and the UK, are routinely testing for viruses for producers and retailers, like Marks and Spencer's. And this is leading to enhanced safety because when positive results are obtained, questions are asked of suppliers around how contamination has occurred. So other pros around this is that it can be used in outbreak investigations, and it is providing data on baseline levels of viruses in food and being used to support decisions around acceptable levels and whether viral standards should be introduced. It's also important for epidemiology and determining what strains are involved. But like all methods, there are also some drawbacks. There's no extraction method for processed foods at the moment. The very large number of controls in the methods means that it's costly. Viruses in foods are not evenly distributed, and this is particularly so for fresh produce, where contamination can be quite patchy and difficult to capture in sampling program. And PCR, most importantly, doesn't distinguish between infectious and non-infectious viruses. So how should regulators and industry interpret results? For every infectious virus genome, for every infectious virus, there are a number of non-infectious genomes. So on the left here, we have an intact virus that's capable of infection. Then we have a virus that's capsid is damaged, not capable of infection. Then we have a virus that has incomplete nucleic acid, which is not capable of infection. And we even get naked nucleic acid. So what does a positive result mean when we're just detecting small fragments of the nucleic acid? Does it mean there is a risk? And that's a question that regulators and industry grapple with. So what's the status on cell culture? It's available for some hepatitis A strains, but it's not easy for routine detection. There has been a breakthrough for norovirus, and in 2016, Itiyabi developed a stem cell-derived human enteroid cell culture assay for norovirus. It's complex and difficult. It is in place in some other labs around the world now, but it's not amenable to routine use. There has also very recently been published a zebrafish larvae assay, and according to scientists in the field, it is more simple and perhaps will provide greater opportunities for research around the evaluation of effectiveness of control strategies. How effective is HPP if we use it on oysters, cold plasma, disinfectants, etc., etc.? These studies are now possible to pursue. So looking at norovirus prevalence, and there has been a lot of work done in many countries now to establish what proportion of shellfish contain norovirus. So this just provides a snapshot of those studies, and you can see that is highly variable prevalence from around 2 to 76%, and that's likely to reflect local conditions. So Australia, for example, is blessed by having a very pristine marine environment in general, and so it had a very low prevalence of norovirus. And the UK, on the other hand, has a very, very dense population and has been found to have quite a high prevalence of norovirus, both in the growing area and at the point of sale. So the overseas data are not particularly informative on what the situation might be in New Zealand with respect to prevalence. There's also been a number of studies on the prevalence of viruses in fruit and vegetables internationally. So Korea, Belgium, France, Canada, Greece, Turkey, Malaysia, Mexico, Australia have investigated norovirus and hepatitis prevalence. And again, the detection rates vary from 0 to 62.5% for vegetables and from 0 to 50% for fruits. This approach can reveal where there are issues at production, processing and retail, and some studies have found problems at particular points in the supply chain and is allowing continuous improvement. So what are the control options? So as I mentioned, for the produce sector, the key sources here are workers' hands and contaminated irrigation and washing water. And this is not rocket science around control. It's really centres on careful selection of water sources, avoiding ingress of sewage contaminated water, evaluating areas for fecal pollution and considering interventions, hygiene training, provision of appropriate toilet and hand washing facilities, and compliance with GAP. And I think here that one point I'd like to make is that guidelines around how industry should operate are essential, and recently in 2019 the Fresh Produce Safety Centre has produced an excellent set of guidelines for the fresh produce sector on food safety, and it covers many of the issues I've just discussed. I'd just like to talk a little bit about Ireland, and I mentioned that I did a little bit of work when I was in the UK for Ireland around viruses in fresh produce. And despite the fact that Ireland had a number of guidelines, they did some hygiene inspections in 2014, and they looked at 131. Could you please mute your system?
Yeah, it's a pump that drains the water, but we'll have a look. If you just have a look, the mic just needs
I'll just plow on, guys.
If someone's got their phone, they haven't got their phone on. I think most people on the call are on mute.
I'll just plow on, Vivian.
Is there anyone, is it coming from where you are? Oh no, there's a couple of phone calls. Can you please put your phones on mute?
Yeah.
Can you please put your phones on mute? We're getting a lot of feedback.
That's better. Nice work, Vivian. Thank you. So with respect to Ireland, despite the fact that there are guidelines there too, in 2014 there were a number of inspections of primary producers of fruit and vegetables, and they found some pretty serious non-compliances. There were actually 3 really serious non-compliances, and 4 FBOs or food business operators were closed. 20% of water samples were non-compliant with E. coli standards. And so I just wanted to put that up because it highlights a bit of a gap, I think. And whilst you can have these guidelines, I think we really need another layer in there, which is around training programs covering gap and explicitly discussing the role of viruses in foodborne disease and the transmission pathways. So other control options for viruses and produce include compliance testing, and certainly EFSA have recommended that there is surveys of norovirus at specific steps in the food chain for leafy greens, tomatoes and berries. Vaccinations of food handlers, probably quite difficult to implement in reality, but it remains a possibility for hepatitis. Increased sanitizer concentration and wash times. Probably it may knock norovirus back a bit, but it's not going to be adequate on its own, and preferably it would be preferable to produce these foods without norovirus contacting the product at all. And there is always consumer advisories. However, as I noted in the rugby match example of the outbreak, these advisories are not always heeded, so it's much preferable that the product is bulletproof at retail. How about shellfish? How do we reduce risk from shellfish? And again, it's not that difficult really in theory. We close areas when things go wrong, improve treatment of waste and effluent from boats, on-site systems, septic tanks, communal systems. Ideally prevent waste from entering aquaculture areas. And I think probably this is a hard one, controlling development of populations and aquaculture areas. So, you know, is there scope for more aquaculture areas to move offshore away from populations? And is there scope to not allow populations to grow where established aquaculture farms exist? And again, it's imperative that these products are bulletproof at retail. And that's primarily due to a lack of steps that can be undertaken to inactivate these viruses once they're on the product. In 2013, this report was produced, and it was by Brenda Hay, Dorothy Jean McCoubrey and Anthony Zammit. It was a Trans-Tasman initiative that looked at case studies of oyster growing areas which were implicated in norovirus illness events. And just a couple of findings from this work that were really important. It was found that septic tanks were repeatedly implicated in these events, or failure of septic tanks, and it was considered that there may not be enough expertise to really assess the efficacy of wastewater treatment systems and on-site sewage management systems. I'm not sure if this has been addressed in New Zealand yet, but it certainly was highlighted as an area of weakness in this report. It was also noted that the annual reviews in shellfish areas that are conducted are not frequent enough, and this is due to development occurring between reviews. And lastly, it was noted that E. coli fails to predict the risk of contamination and that programs should be supplemented with viral testing. And on that note, around viral testing in particular, I just wanted to touch on international initiatives. So there has been work done by Codex, and there is now a Codex guideline that recognises the need to do some viral testing in some situations, certainly following outbreaks and rainfall, etc. We all await the publication of the US FDA, Health Canada, CFIA risk assessment on norovirus, and this aims to set performance objectives or levels at retail and food safety objectives, levels at consumption for norovirus. You also might be aware of the European Union work, which has involved surveys, extensive surveys of norovirus throughout Europe, which have provided information on the extent of norovirus contamination. This was explicitly requested through the European Commission to support the development of norovirus thresholds. So it's a really interesting discussion. This is these are some results from 2012 in Europe, which show the proportion of oyster samples with norovirus in the quantities. So the green has norovirus at less than 100 viral genomes. The darker colours go up to very high levels. So you can see for the UK that a significant proportion of oysters are contaminated at low levels, and during winter we get much higher levels. In France, contamination in general is much lower. I guess the table below highlights the possibility of failure if such a system was introduced. And so you can see if the regulatory level was set very low at 100, we would have around 33 to 83% of samples failing. And so the impact on industry of these will be significant if introduced at a low level, and at a high level perhaps less significant. Whether it heads that way or not remains to be seen, and I'm not sure whether New Zealand yet has a handle on what the impact of such standards would be here. So finally, in considering the future and how we reduce the risk of illness from viruses, a few last thoughts to emphasise. The cost to government and industry and wider society from illness outbreaks is very high, and this relates to healthcare costs, job losses, reduced consumer demand for products, and flow-on impacts such as reduced tourism. Preventing an outbreak is far cheaper than recovering from one, and it's critical that food businesses are vigilant and understand and follow best practice. There is a need for increased education and training, particularly around GAP and the role of foodborne viruses and disease. In relation to shellfish particularly, we need to focus on ensuring sewage treatment is as good as it can be, and that septic tanks are designed appropriately and are inspected. And finally, most countries are analysing food systematically to understand prevalence at different points in the supply chain, to gain a better understanding of where viruses are being introduced, at production, at processing, or at retail. And there is a move to introducing viral standards in foods. How well does New Zealand understand viral prevalence here, and what would the impact of such a standard be? So lastly, New Zealand has a really great opportunity. It has been estimated that 40 million people consume New Zealand food. We've got a growing global population, and New Zealand is blessed by a relatively low population in fertile, productive land and sea. We need to protect this resource and our reputation as producing safe food. So think globally, but we need to act locally, be vigilant, ensure water is clean and hygiene is scrupulous. So thank you for listening to me today.